Hi, I'm Roxanne Newman, Senior Conference Producer for Technology Networks, and I'm here to introduce our next talk, Key Factors to Accelerate Drug Discovery. I'm really pleased to welcome Dr. Pei Liu as your presenter today. Pei received her PhD in pharmacy from Nanjing University in 2019. She currently works at GenScript as a senior scientist in the Reagent Antibody Department. Her work is mainly engaged in antibody development and validation. She established a single B-cell platform for antibody discovery and won many honors for her major contributions in the discovery of COVID-19 antibodies. In addition, Pei has worked with various cases and has accumulated a wealth of experience on the development of ELISA kit. Now, using innovative technologies, she is committed to accelerating the development and application of biopharmaceuticals. Following the presentation today, we will have a Q&A session and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during today's presentation. You can also view all the fantastic supporting literature from GenScript today, which you can download for free below this live stream panel. Lastly, if you experience any technical difficulties during the presentation, please request help using the technical help icon in the menu on the left-hand side of the player. Now, without further ado, I will hand over to Pei to begin her presentation.
Okay, thank you for the introduction. Hello, everyone. This is Pei from GenScript. I'm very happy to have this opportunity to discuss with you about the key factors to accelerate discovery. We will go through key steps in drug development process, then comprehensive and advanced antibody generation platforms. Another important part is important keys to accelerate drug discovery. We also review GenScript technical advantages. So from the business perspective, GenScript Group currently has 4 subsidiaries in 4 directions. The largest and oldest is the Nanjing GenScript, is mainly engaged in R&D outsourcing service in the field of life science. And the ProBio is committed to building a comprehensive and complete CDMO platform to accelerate the drug development. Benzyme Engine is doing industrial admins and the legendary currently cooperating with Janssen focusing on the cell therapy. So the first R&D outsourcing service, we committed to providing customers with one-stop R&D outsourcing service from molecules to peptides, proteins, antibodies, and cells. In terms of the protein, our main focus is customized protein production. The 4 main platforms, including prokaryotic expression of E. coli and bacillus and eukaryotic expression and purification of the insect and mammalian. At the same time, we also undertake lentivirus package lentivirus service. So the most important in terms of antibodies, we have 17 years of antibody preparation experience. ALAC and OLAW internationally certified animal facility and production through up to throughput of 1,000 projects per month. Many help customers to do polyclonal antibody, monoclonal antibody production, in vitro detection, monoclonal antibody development, and test kit development, including PC kit and HCP kit. Based on our in-depth understanding of the entire process of antibody drugs and protein drug development, We also have in-depth knowledge and rich experience in the development of PK and HCP kits. In addition, we also undertake cell engineering projects, including overexpression cell lines, reporter gene cell lines, also the CRISPR-based knocking and knockout cell lines, and the protein antibody production cell lines. Here, I think everyone is very familiar with the drug discovery and the development process. From initial discovery to a marketable medicine is a long, challenging task. It takes about 12 to 15 years from discovery to the approved medicine and requires an investment of about $1 billion. So the process involves the identification of the candidates, synthesis, characterization, validation, optimization, screening, and assays for therapeutic efficacy. Once a candidate has shown its significance in these investigations, it will initiate the process of drug development earlier to clinical trials. From this process, everyone can also find that drug discovery and the preclinical studies account for the heaviest portion of heaviest proportion, and the time consuming is the longest. So only the pre-stage work is done well enough that the subsequent process can be carried out smoothly. So how to get a promising candidate? The process is sufficiently long, complex, and expensive so that many biological targets must be considered for every new medicine, ultimately approved for clinical use. And also research tools may be needed to investigate each new target. Successful R&D and preclinical development is dependent on quality reagents for detection, characterization, and quantification. So how to guarantee the success of the drug discovery process? So the important factor is first to have an excellent technology. First, a well-designed antigen could be ensured that we can obtain the positive antibody against the target. So the design software can solve this problem, and that antigens are difficult to express or can't be expressed. Optimal and also the optimal antigen design tool have been used for more than 5,000 projects, and the success rate is more than 98%. After the antigen design, the second critical step is the immunization strategy. The ImmuPlus technique can break immune tolerance and used on project with high homology to mouse. And also we have adjuvant library, which are very helpful from different immunogens. The mono-boost technique only needs 2 weeks for immunization. So the third key step is antibody screening characterization. We improved the traditional hybridoma platform by using the electrofusion system with extremely high efficiency. The advanced B-cell cloning and beacon platform could shorten the timeline for screening with more positive hits. After screening positive hits, we needed to produce more antibodies to validate its function with multiple assays and cell lines. Here is a comparison between the ImmuPlus and the regular immunization group. The immunogen is 100 homology with mouse, and the animal response is very well when we use the ImmuPlus. And finally, we got several FACS-applicated positive clones. ImmuPlus has also successfully used to generate antibodies against the antigens from same species, which have greatly utility in specific reagent antibody generation and drug validation and development. As we know, loss of drug targets are CD moleculars or multiple transmembrane proteins. For single transmembrane proteins, it must be easy to express the protein in vitro and use it to immunize the animals. For example, the 421BB protein. However, it's hardest to express the multiple transmembrane protein. DNA immunization is a very appropriate method for multiple membrane protein. For example, the Claudin 18.2 is a 4 times transmembrane protein. We got a good results by using our DNA immunization system. Our DNA immunization system have 2 advantages. First, we optimize the vector, which could be improve the protein expression level in vivo. And second, we could immunize the animals without using the gene gun. So until now, hybridoma technology is still widely used in drug discovery. By using the innovation technologies, could be improve the success rate of the project. And here I want to highlight our more advanced rabbit monoclonal antibody platform and the Beacon single B cell platform. For rabbit monoclonal antibody platform, first we will separate and enrich the PBMC to obtain antigen-specific B cells, screening positive clones after stimulation and culture, and finally obtain positive antibodies through sequencing and recombination. Since its establishment in 2019, the rabbit monoclonal antibody platform has successfully carried out more than 200 projects. On average, more than 50 positive clones have been obtained in each round. The unique Phoenix monorab could got antibody in 11 weeks. And also for the single B platform, we could obtain the positive clones in 1 month, and currently available for mouse and rabbits. So a comprehensive screening system is also an important factor in obtaining target antibody. We not only have various ELISA methods, but also cell-based and FACS screening method to ensure that the screened clones can recognize the naturally expressed protein. So after we got the positive antibodies, it's very crucial to invalidate the performance of these antibodies, such as the affinity, epitope, and the blocking function, whether it has cross-reactivity, and so on. Here is a case for the Claudin 18.2. We use DNA immunization and the hybridoma platform to obtain several positive clones. Immunization strategy is the first is the cofactor plasmid will be injected into the mouse 3 days before immunization. The gene immunization was performed 7 or 14 days apart. Through this immunization plan, we obtained mice with beta titers for fusion, and finally obtained positive clones by FACS screening. In addition to this pure genetic immunization strategy, in the design of other projects, we can also integrate whole cell protein or VLP into immunization, and finally get antibodies with better immune response. This case was done on B-cell cloning platform. Now rabbit antibodies are popular. FDA have approved the 2 rabbit antibodies drug. One is Bruluzumab, a novel anti-VEGF humanized single-chain antibody fragment for treating wet AMD. Another first full-length rabbit-derived monoclonal antibody drug is anti-CGRP antibody, and the trade name is VAPT for the prevention of migraine.